sound check. Looks like we're good. Okay, so today we're going to focus on the theory behind the subparts of a cell. We've talked about cell theory already before. Cell theory basically states that the cell is the smallest functioning unit of life. Is there stuff smaller than cells? Yeah, but they can't survive on their own. Like, they're now dead. They're not alive. Like, a nucleus on its own is no good. Cytoplasm on its own is no good. You need to have all of the parts of a cell put together for it to be considered alive. And so we're going to focus on this word. And you know how at the beginning of the unit I suggested you might want to have like a definition sheet, perhaps? If you made one, maybe grab that today and let's write down some of my bolded words, because today is one of those like I'm telling you names sort of day. So here's the first bolded word I want you to know. It's called an organelle, and I believe the word came based on the idea of, you know how like as humans, we are alive, but we are alive because we have many different organs in our body, like for example a brain or a heart or a kidney. Right? Now there are some organs you can live without. For example, you don't need an appendix. It's not really necessary. My wife had her gallbladder removed. You can live without some organs. But like, you can't live without a heart or a brain. Yeah, you might be able to get away with a part of a liver, but like you do need a liver, at least some of your liver, because it filters like toxins out of your body. Yeah. Now you can have a liver transplant, or you can have part of it removed. You might not have the entire part. Yeah, you're gonna need a liver. You die, or you need to do dialysis to like filter your blood out of all the toxins out of it. So. Anyways, we're getting off topic here. You guys know how your body has organs in it? Okay. Well, just in the same way that a body is made up of smaller pieces, organs, a cell is made of smaller pieces. We call them organelles. Okay, two more vocabulary words I want you guys to divide things into. The words are eukaryotes and prokaryotes. And the definitions are like literally right here, so I'll just read them off here. If it's a eukaryote, it means that there is a nucleus bound in a membrane. Okay. It means that you have a nucleus, and the nucleus itself has like an outer protective coating on it. Prokaryotes, then, are things that they don't have a nucleus. There are some cells that actually don't have. You guys know what a nucleus is, by the way? Yeah, but like, what does it do? What is its function? What's the analogy of a nucleus? Nucleus is like the brain of the cell. Yeah. And so there are some cells that actually don't have a true nucleus. Good examples of this would be bacteria and viruses, which were some of the things we were talking about in previous lessons, where scientists, they didn't really know about bacteria. They just thought that spontaneously things grew, right? But now that we know more about viruses and bacteria, because we can look at them under microscopes, we can basically categorize our cells in one of two things. Either it has a brain or it does not. Fair enough? Okay. Give you guys a few seconds just to write these down on your definitions list if you're doing that. Eukaryotes and prokaryotes. Most of my next slides are basically going to be a combination of pictures as well as the definitions and kind of what we believe they look like. So here's a picture of kind of a generic cell. And if you're doing my project, this is why I want you to do some sort of like three-dimensional part to it rather than just a flat picture, because maybe the three-dimensional bit kind of gives you a little bit of an idea. So let's start here. This is the nucleus. And the nucleus is usually considered to be like a ball within the cell. Okay? Usually it's like a marble or a jawbreaker or a gumball that kids will use when they design these cells. Let me give you an analogy if you want to use Lego pieces. The group that did Lego made their nucleus City Hall. Because what does the nucleus do? It directs all of the activities of the cell. It basically tells everything else what to do. It's the boss. It says, you do this, you do this, you do this. It's my bossy 6-year-old telling her little sister what to do. Does that make sense? So it's the brains of the operation. That's your nucleus. Okay. You guys should have heard of that one before. Okay. What about mitochondria? Is that a term you guys have learned before in grade 8? I don't know. It's just what it looks like, I guess. Do you know what a mitochondria does? What does it do? Anybody know what a mitochondria does? It's a power source. Yeah, yeah. It provides the energy for a cell. So in terms of an analogy, again, if you like the analogy of building a Lego city, this would be like a power plant. If you had a power plant, that's the mitochondria. So you really, all cells that are in the eukaryote family that have a nucleus, they really need those two things first. You need to have a brain, which tells everything what to do, but then you need to have the power source to give it the energy so that it can do stuff. Okay? So those are usually two of the first things that kids have learned about with cells. Okay. Another one you may have heard of before is the membrane. And maybe not. I'm not sure what you guys remember from grade 8. But a membrane is kind of like the outer coating, the outer surface. Let me give you like an example of this. You guys see my white table right here? Okay, the top surface is white, but there's edging around it. You see this brown, this gray edging right here? That would be kind of like a membrane in a way, where it's just around the outer surface and it kind of keeps everything inside of it. Does that make sense? And so if I wanted to give some other analogies, let's say this was a city or a town, perhaps there is like a gate around the community. I don't know. That could be like a membrane of some type, right? Where it basically divides what's either in the cell or what's out of the cell. What happens if a cell membrane gets punctured? It literally, like, the guts of the cell would start to like spill out. Yeah, right? So the cell membrane, maybe another analogy is it would be like a balloon, and there's stuff inside the balloon, but if you puncture the cell membrane, then stuff can start gushing out of the cell. So I had one group then that made their cell, they made it in a big bowl, and they used Jello, and the bowl represented the membrane. They used like a clear plastic bowl, and so that's what kept everything inside. If it wasn't for the bowl, it would just start spilling everywhere. So hopefully those are a couple of the words you guys have heard before. Maybe one more you've heard, and then I'll skip to new slides. Cytoplasm. Anybody ever heard of cytoplasm before? Yeah. Anybody remember what it is? It's kind of like, you know how I use the analogy of using the Jello mold? Well, the Jello is the cytoplasm. Maybe here's a different analogy. Anybody ever buy something from, like, say, Amazon, and it needs to get shipped to you? And it's a little bit fragile, and so they put it in like styrofoam, or they put like those bags of air inside to kind of keep it from like rattling around. That's what cytoplasm does. It's kind of like the filler, so that all of these other things that are found inside the cell, so they don't just go rattling around everywhere, they're kind of like soft and cushioned. Does that make sense? All right, let's go to my slides here maybe, because I'm not sure how much of that you guys knew already. Let's talk through these ones here one at a time. So first one I've already discussed is cell membrane. What does it do? It is a protective barrier that keeps things either in or out of the cell. Right? It's the outer coating. The nucleus is usually the largest of all the organelles because it has the most responsibility. It's headquarters. Any of you guys ever watched the movie Inside Out? The one with the little Disney, the Pixar movie with the girl who has all the people running around inside her head, the 5 different characters, right? Like headquarters, if you see that movie, that would be like the nucleus, right? Where all of the activities are directed from. Okay. And then cytoplasm is the gel-like substance inside the cell membrane. That's like the cushioning. It's like the styrofoam in packaging. It's like those bags of air that you might put inside of things, or like the bubble wrap when you ship something, because you don't want it rattling around all over the place. So it's a gel. Okay, the next two things that I want to talk about here are actually only found in plants. Only found in plants. So make sure that's clear right here. This is plants. That's pretty good for sideways. Okay, plants have two organelles unique to them. Let's talk about the bottom one first. It's called the chloroplast. Maybe you've seen this at some point before. We're actually going to watch the Magic School Bus one day when we talk about plants. Yeah, classic Magic School Bus. Chloroplasts are basically the organelle in plants that lets them do photosynthesis. What's photosynthesis? Well, it's where plants take water, carbon dioxide, and sunlight, and it makes them sugar and oxygen. It's how plants feed themselves. If we as humans could ever put chloroplasts into our DNA, that would be incredible. Well, yes, first of all, we'd all be green, which would be a little weird, but we wouldn't need to eat anymore. We could literally just go outside and go, and that would be eating for us, right? Like the world's food problems would be solved. The world's carbon dioxide problems for global warming would be solved because we would literally go breathe in some carbon dioxide and we'd be eating. We'd have to drink water. Yeah, exactly, right? You'd be outside in the sun, and you'd be like, I'm full, I better go inside. But we'd all be green, by the way, because chloroplasts are green colored. So. Oh, we're talking like genetic engineering, right? Because we'd have to find a way to take human DNA, which doesn't involve chloroplasts, and somehow try to find a way to splice that with like plant DNA. So I don't imagine that's close, but that'd be cool, wouldn't it? Anyways, here's the other one that's different. It has a cell wall, and this is why this is really important. As humans, when we die, our bodies will decompose. Okay. However, what happens to trees when they die? They can still be used as wood in construction projects. This is wood right here, right? And the reason why this piece of table here hasn't decomposed is that it has cell walls to it, which is like a frame around it. It's almost like a cell membrane. Almost like a cell membrane. The better example, though, would be this. Take humans, okay? Humans, our cell membrane is our skin. And if I cut my skin, blood comes gushing out of me, right? But the cell wall to humans is our bones. Does that make sense? So the skin is the outer surface of a human, just like the cell membrane is the outer surface of a cell, but there's something inside of it propping it up, like a frame. Here's another analogy, a tent. Everyone ever go tenting before? Okay, right? The actual fabric of the tent would be the membrane, but like those stick type things that you have to like bend and twist to make like the frame of the tent, that would be the cell wall. Here's why this matters then. When a plant dies, the cell membrane will maybe eventually fade away, but the cell wall, the bones, the structure, the framework stays alive, and that's actually really useful, which is why wood still has like hard, rigid properties to it, even after the plant is dead. Really, the only reason why that stops then is maybe bacteria or molds or funguses eventually like rot away a piece of wood, like, you know, a log in the forest. Like, okay, that can eventually decompose, but that's because eventually it's being rotted by bacteria. Does that make sense? So, anyways, these two are unique to plants. We as humans don't have these. Yeah. Okay. Here's the next one: vacuoles and vesicles. Okay. These are storage places. Okay? So, if you want another analogy, a human body has a few different storage places. For example, one might be, say, our lungs. Another might be, say, our stomach. They're temporary storage, because the stuff that we put there doesn't necessarily stay there the whole time, but like our lungs are meant to store oxygen and carbon dioxide. Our stomach is meant to store food, right? And I mean, it moves on, right? But basically, you need that same sort of thing inside of a cell. And basically, this is like some sort of little bubble that you might find inside the cell, and this little bubble, you can put stuff in, food or oxygen or whatever proteins you might need. So vacuole or vesicle, there's two different words for it. I believe the difference is one of them is long-term storage and one of them is short-term storage. I always forget which one is which. All I need you guys to know is it's all about storing stuff. Okay. Actually, let me elaborate a bit more. It can also store waste. Okay. As humans, we need to go to the bathroom, right? There are human body function places that store human waste, and eventually it leaves. And in the same thing, like plants have that too. Animals have, like our cells do that as well, right? Where it needs to get rid of certain things. So it's temporary, or it's either short or long-term storage. Okay. Here's some terms then that you may not know from grade 8. Okay. This one's called an endoplasmic reticulum. And it's basically tubes that materials get transported through. Have you ever been to Costco and seen how they have those suction tubes by the tills where they put some money or some stuff into that tube there, they put it in and then it goes sucked through. You know what I'm talking about? Okay, well, for those of you who've been to Costco and seen that, you'll get the reference. I mean, it's like the analogy would be like the postal service, where it's all about transporting things through like a series of tubing. If I wanted to use the human body as an analogy, what sort of tubing do we have in our body that stuff gets transported through? Intestines will work, yeah, that'll work. Veins and arteries, yeah. So think of like in the human body, anything that's got like a tube-like sort of structure to it that you can move things in, that's endoplasmic reticulum. And it often works in correlation with your vacuoles and vesicles, because this is where you store the stuff, and then when you're ready to move it, you send it through the pipelines. Okay. Hypothetically, let's say you want to use the Lego city, like my one students did. What would the analogy in the Lego city be then? There's a few different ones you could use. Roads would be good, yeah. Yeah, or if you could somehow go underground and have a subway, that'd be kind of cool. Okay, ribosomes are another one that you probably haven't heard before. Ribosomes are usually attached to the endoplasmic reticulum, and they are where proteins are made. Okay, your body is made of a lot of different proteins and lipids and carbohydrates, and like there's lots of different things involved in your body. When we need to synthesize proteins, that happens in ribosomes. Now, I don't need you to know how that works and all the little details of this. If you want to know that stuff, take Bio 20 and 30. All I need you guys to know is ribosomes are about proteins. Okay? And they're making them. Because the opposite, in a way, of that is lysosomes. And lysosomes destroy and clean up and basically remove unwanted things. Little trick here. Here's how I remember lysosomes. Lysosomes is almost like the word Lysol. What do we use Lysol for? It's like a disinfectant. It's a cleaning agent, right? We use it to scrub down sinks or toilets, or right? If you've used Lysol before, right? And so it's meant to destroy germs and kill bacteria. That's what it does. These are membrane-bound sacs filled with enzymes, and they kill things. If you have an invader like a germ or a bacteria, or if you ever have a damaged organelle, it breaks them down. Okay. In a way, lysosomes are kind of like your stomach in another way, because what's inside your stomach? Food, but yeah, and the acid dissolves and breaks down the food. Does that make sense? And so that's kind of what the lysosomes are like, is their job is to destroy and break down things. Okay. Now, let's say that it does, something in your cell gets broken down, and it needs to get removed. Well, let's go back and do a little bit of a review. Where do we store it? We store it in a vacuolar vesicle. How do we move it? We move it in the endoplasmic reticulum. How do we get it somewhere, though? We have something called the Golgi apparatus. And what these things do is that they form vacuoles by basically, maybe this picture can help make sense. Can you guys see how there's like this little disc right here? And there's like another little disc right here. Imagine that this right here is a little bit of waste that needs to get removed. What's going to happen is this thing is going to like wrap all the way around it. It's almost like a piece of paper. Let me do a demo here. Here. There's this little sack right here. Okay. And here, this is some waste. What's going to happen is it's just going to wrap around it, and now it's kind of like in a bubble. The bubble then would be the vacuole, and it came from the Golgi apparatus, which basically engulfed it and circled it. Okay, say that again? Well, so the thing inside, this could be food, this could be waste, this could be oxygen. I don't necessarily know what it is, but I need you guys to know that these two things, really these three things, this one and this one, they work in tandem. So the Golgi apparatus basically is like the item that like turns into a vacuole, and then the vacuole travels through the endoplasmic reticulum. See how that kind of works together? So they're flat discs that then form a bubble, and then you can then, usually these things are for removing things from the cell. Okay. The analogy that some people have had is this is like a shipping company. It's their job to take all of the bottles, put them into a box, right? And so the packaging company puts everything into a box, and then the shipping company would be like the endoplasmic reticulum, which then like sends it out on a truck. Last one, mitochondria. We've already talked about that a bit. How do all of these things even run? Well, they need energy. And that energy, I think I have it on a future slide. I want to make sure I have that acronym on there. No, maybe I don't. Okay. There's a little 3-word acronym I need you guys to know here. The energy actually has a more specific word than energy. It's called ATP. It is adenosine triphosphate. And it is the chemical that is stored that provides the energy. It's adenosine triphosphate. I'm sorry I didn't write that very nicely. You don't need to memorize the name here, but you should know that ATP is the 3-letter acronym we use to describe energy. Adenosine triphosphate. Okay. Long story short, mitochondria is like the power plant. It stores all of the energy that you need so that the cell can then do all of this stuff. Okay, so I want to go back to the slide here and just do a quick recap then. So now that we've talked about these things some more, see these Golgi little things here? You see how like this thing right here, it's like half of a circle right now? It's basically just going to keep making a full circle, and if it does that, a Golgi complex then turns into a vacuole. And a vacuole is where you then store something for a little while, right? Let's say that you want to move it from one place to another. You use the endoplasmic reticulum, which is like these little pathways, right? And these pathways allow things to basically move around like a roadway system, right? Lysosomes are very similar to vacuoles, except vacuoles are for storing things. Lysosomes are for destroying the stuff you don't want. It would be like the trash compactor, or anyone have like a garburator in their sink where you flip on an extra little thing and it chews up all the food before sending it down the sink? Right? That'd be like a lysosome, right? Ribosomes are attached to the endoplasmic reticulum, and they make proteins that we need. Cytoplasm is all the gel in between. It's like the filler, so that it doesn't get rattled around too much. I think I've covered everything otherwise. So, does that make sense? Okay. So let me go back to your project for a second here. Now that we've talked about all of these organelles here, if you like the idea of this project, and I hope you do, because it's usually kids say it's kind of fun, right? You can steal ideas that I've given you. You could make a Lego city, and the analogies would be like City Hall is the nucleus, and the post office. What would the post office be? What does a post office do? It gathers things together before it decides to ship them out. What would that fit best as? Golgi apparatus, yeah. The roads then could be your endoplasmic reticulum, right? What about lysosomes? What would be like a city analogy for lysosomes? Yeah, like a garbage dump or something like that, yeah, before maybe it gets incinerated. Yeah. So you could make a Lego city if you want. Another group, like I said, they just baked a cookie, and they just put random things on the cookie that kind of looked, to the best of their ability, like, where's that slide again here? There it is. That looked kind of like this. So they put on gummy bears, they put on, I think they used licorice for all of the different tubing, if that makes sense. They found that licorice that you can like tear apart into like little strips there, which is kind of cool. Yeah, right? They used like gummy bears and jawbreakers and little candies, right? And they made a really big cookie, and then we ate it afterwards. So that's kind of cool. So anyways, that's your project if you choose to do that one. So, let me keep moving here then. I've got a few more slides here just discussing vesicle and vacuole. Oh, I do have this. Okay. Remember how I talked about there's short-term and long-term storage? Okay. The vacuole is long-term storage. A vacuole is a cell which is not meant to move around the cytoplasm. It's basically you're going to keep stuff there for a really, really long time. The vesicles, though, the vesicles are the ones that like are designed to be like, I don't know, I think of like a storage pod that like can somehow like move through those tubes, if that makes sense. So this one's designed for moving, and this one's designed for just sit there. Right? It would be like what's that place up on some of those places up on the north side or on the far end of the west side, like those storage places, those big what do you call them? I don't know, storage places, I guess, right? Storage lockers, right? Where you can pay $100 a month and store your random stuff that like your wife won't let you keep in the garage. No. Anyways, okay, let's move on here. I've got another slide here talking about some differences between plants and animals. So we've already discussed a few of them, but make sure you're clear on them. Okay. Plants have a cell wall, animals don't. Why? Well, because plants, this is really useful. After a plant dies, it still has like some rigid strength to it. A great example is bamboo. Bamboo is a plant that can grow, but like once you cut the bamboo off of the ground, it's one of the strongest and most flexible types of wood that we know of, right? It's actually quite useful, especially in like South Asian communities, right? If it wasn't for the fact that there's like that cell wall framework hidden inside those cells, it would just like fall apart, right? Like we as humans eventually do, right? You know, when we die, we decompose. Chloroplasts we discussed as well. The idea that plants plants do have them, we don't. Okay, here's some ones we haven't discussed. Plant cells usually have very, very few vacuoles, and they're massively large. Let me show you a kind of a picture rendering here. This little white thing right here is probably a vacuole, a little storage pod. Plants usually have one big, massive one. So rather than having a whole bunch of small ones, plants just have like a big one. The analogy I might give for that would be like Amazon headquarters in Calgary, or like they have these massive warehouses, and in this big warehouse they have like everything, and they have like one big warehouse. Does that make sense? Whereas then the stores in Lethbridge and in Medicine Hat and Pincher Creek, there's a whole bunch of like smaller storage containers there, right? So like there's a Brick in Lethbridge, there's a Brick in Medicine Hat, there's a Brick, you know the Brick store, one where you buy couches and TVs from, right? Right? Do they have everything in their store? Well, no, but the one in Calgary, that's probably like a big super center, right? So that's the analogy I use. The other one right here is lysosomes. Animals have lysosomes, plants don't. Which is one of the reasons why humans and plants, we have an immune system. We have an ability with white blood cells to kill invaders. You ever heard that before? White blood cells are cells that are specifically used to like protect our body from disease and invasion. Plants really don't have that, which is why we use things like fungicides and insecticides on our plants to try to prevent our plants from being killed by germs and infestation, right? So we have an immune system. Okay. There's one more thing on here that I haven't discussed. It's something called the centriole. Okay. Centrioles, since I didn't have a previous slide on it, they're in charge of cell division. And what I mean by that is these are organelles that control mitosis or meiosis. You guys remember doing that in grade 9 a little bit? Okay. If you were to break your leg doesn't stay broken because your body will make new bone cells to heal, right? And so as a human and animals, we have the ability to have like some degree of regenerative growth. There are some animals that actually have a high degree of that. If you cut a tail off of a lizard, it can actually grow its tail back, which they actually did in the Spider-Man comic books, by the way. Anyways, side note. But like plants don't have that ability. Okay, plants do grow, but like if you were to like snap a plant in half, it stays broken. right? But if you were to break your leg, if we reset your leg, it can actually like refill in the gaps, and you'll actually make new bone cells or new heart cells, right? So anyways, through that process here, we have centrioles, which allow for cell division. Okay. Here's a few more pictures. I'm not going to regularly be asking you to identify what stuff is inside of a cell, because like it kind of looks like gibberish sometimes. Like I imagine this could be a vacuole or maybe a lysosome. I don't really know which one it is. I know for sure what the tubing is and what the nucleus is, but like some of these items, you don't have to know exactly what they look like. I just thought I'd give you some pictures though, in case you want to use this when you're making your physical model. So yeah. Yeah. Yeah, you should definitely have a legend on your project so that I know what is what, right? So if you're using, say, licorice, make sure that you write me and tell me licorice is the endoplasmic reticulum, and gummy bears are the mitochondria. Oh, oh yeah, you have to like just describe them, you mean? Yes, yeah. Let me go back to that project here. In terms of the marks, you get 2 marks for making the organelle, and then you get 3 marks for a legend and describing what they do. So I want you to describe and tell me, I am using City Hall as my nucleus. The reason I'm using City Hall is that City Hall is what tells everybody in town what to do. It directs the activities, right? So yeah, make sure that you don't just make it, but that you describe why you did what you did. Good question. Okay, I think I'm almost in my notes. There's only, yeah, there's a little bit left here. So I've got some pictures here, but I think I've made my point. Is everyone fairly clear on like some of those sub parts of a cell? No questions on that? Okay, last part and then we're done. I want to spend a little bit of time talking about the cell membrane. It's actually one of the most important parts of the cell because it's the gatekeeper. It's what lets things in or keeps things out, right? It's the outer bubble, so to speak. Okay, the cell membrane is actually a very complex item. It's actually not one thing. It's actually many different little things put together. And it has a bolded word I want you to know here. It's called a phospholipid bilayer. I want to spend a little time talking about what the heck that means. Phospholipid bilayer. And I think the best way of me describing this is to actually go to my next slide, which is a picture of it, and then I'll come back again. Here is a picture using electron microscopes of what they believe the phospholipid bilayer kind of looks like. Okay, this is kind of the way that the cell membrane works. Okay, now if I could draw a little bit better, I'm going to draw over here on the side. A cell membrane kind of looks something like this. Here's the outer section of the cell. What you really have is like a ring almost, right? And this would be outside of the cell, and this would be inside of the cell. Does that make sense? How we'd have to have like a ring almost as the membrane? And if we were to zoom in on the ring, this is what it would look like. So like this might be outside of the cell, outside, and this could be the inside portion of the cell. And basically this is like a barrier, okay? So we call it a phospholipid bilayer. And the reason why we call it a bilayer is that it actually is 2 different things back to back. Right here, I'm going to use a highlighter, I think. Right here, this is like one layer, and this is like a second layer. It's the exact same thing, but like squished together. Again, another analogy, you ever make like a peanut butter and jam sandwich? So you have a piece of bread, and you put peanut butter and jam on this side. And you have another piece of bread, put peanut butter and jam on this side, and then you take the 2 of them, and you put them together like that. That's kind of how this phospholipid bilayer works, is that this is the bread, and this is the bread, but then there's the stuff on the inside, but you get double the stuff on the inside. Okay? So this stuff right here, these are phosphates. Oh, phosphates. And you guys now know from chemistry what a phosphate is. A phosphate involves a PO4 3-. And you also have phosphates down here as well. Okay. And then on the inside, you have lipids. And if you guys don't know much about lipids, lipids are like oils and fats. And so what you end up having then is this sandwich of a phosphate, a lipid, more lipids, and then a phosphate again. And that's what we believe it looks like on an internal level. So I'm going to go back to the slide here again, and this is kind of the same thing here, only maybe it's a better picture here. So this could be like the outside of your cell. This could be the inside of your cell, and there's this double layer here where you have a whole bunch of phosphates on the top, then some lipids on the inside, then some more lipids on the inside, and then the phosphates on the other side. And it's like I said, two pieces of bread in a sandwich kind of coming together. Okay, let's talk about some more properties though. Why is this so crazy intense? It has to do with these two words, hydrophilic and hydrophobic. Anybody ever heard the word phobia before? What is a phobia? Yeah, it's something you're scared of. Now, our cells aren't actually scared of things, but like we use the word to describe things that you either like or don't like. Okay? And so hydrophobic, these are things that water, hydro is the prefix for water, okay? These things do not like water. So the inside of the membrane does not like water molecules at all. And so you should never find water inside of this barrier. You should either find water on the outside or on the inside, but you should never find it inside the barrier. Does that make sense at all? Okay. Whereas the phosphates are on the outside and they do like water. So can a water molecule touch this thing right here? Sure, no problem. Can a water molecule touch this thing right here? No problem. But should you ever find water right in the middle of this barrier? No, which is why it's a great barrier, right? Because what ends up happening is water either stays here or it stays here and it doesn't stay here. So it either keeps it out or it keeps it in, right? Just like, say, a tent, right? The idea behind the tent is that water either stays on the outside or on the inside of the tent. But like the actual barrier itself is supposed to be like protecting you. So we call that the phospholipid bilayer. And sometimes there are proteins inside the phospholipid bilayer, and they're like secret passageways. We're going to talk about this next week on Monday or Tuesday, but these proteins, they go all the way through the bilayer, and literally it's like a secret passageway where when you want water to come inside, they go through this passage. But only if the protein lets you. Okay, we've got a whole day devoted to talking about that, so I don't want to spend too much time on that. But I need you guys to memorize these words then. Hydrophilic, hydrophobic. Okay. Here's kind of a better picture maybe of what's going on then. Okay. The cell membrane has this double layer, and yeah, these proteins there, they're those secret passageways where things can go through if you want. The word we use then is called the fluid mosaic model. And basically the idea is that it's fluid, meaning that like it can kind of move around a little bit. You know how there's that cytoplasm that's like this gel-like component? Because of that, this opening right here and this opening and this opening right here, they're not always in the same spots. They can move around a little bit. And so that's why we use the word fluid, because these things can actually move around. Okay? And there's kind of like a random patterning to it as to where the proteins are. If I could give one more analogy, this would be like the big wall. Okay? I'm just trying to think of a good, maybe if you've watched, you guys shouldn't because it's 18A, but if you watch Game of Thrones and they have like the big wall in that, or you know how Trump wants to build a big wall across Mexico? Okay. If you can think of this thing as being a big wall, these things right here are the gates, right? And there's different gates. Now, normally when we build a wall, the gate's always in one spot, but inside of cells, the gates can kind of move around a little bit. Okay, my last slide here. The word we use to describe this then, that I want you to know, is the word semi-permeable. And if you happen to have a textbook, I don't use it much, but it actually talks about it on this page in a textbook. Semi-permeable basically refers to the fact that it keeps some things in and some things out. Some things can go through and some can't. Let me talk about a tent again. What does a tent keep out? No, not air. It keeps out water. Bugs, probably. But you know what it doesn't keep out is air. Because if air couldn't get into your tent, you would suffocate at night. Does that make sense? So that's why we use the word semi-permeable, because some things can fit through and some can't. Okay? This is a good picture I found here. all these blue things could be water molecules. And water cannot fit through unless you have one of these special gates to go through. Does that make sense? Water can't go through this membrane, but maybe other little particles could, like say air particles. And so it's a semi-permeable membrane because some things get stopped and others don't. Right? So if I go back to this picture right here, these are water molecules. The only way they get in or out of a cell is by going in through the gate. But if I had like a smaller particle, like say oxygen gas could maybe fit right through the gaps there, just like it does in a tent. So, and I think that's my last slide. I know you're probably itching to get out of here, but do you guys have any questions you can think of? Okay, let me give you a quick summary, and then we'll be done for the day then. We've now covered 3 sets of notes, and you can probably start to do some work. So I'm not saying I need you to do homework over the long weekend here, but like in the next couple of days here, start on a few tasks. You can start on the project, you can start on your assignment now. Today's goal was to focus on all the different parts of a cell, and hopefully I've given you a lot of different analogies to what they do and what they're like. Okay. That's actually one of your assignment questions, by the way, since I'm chatting at you here. When I find your assignment now? One of the questions on the assignment here is to explain, here, let's do some of these ones together now. One of your assignment questions says to label and explain the functions of the parts of the cell right here. You should be able to do that now. You should be able to tell me what all these things are and what they do. So here's a picture of a cell. It came right out of your notes, you know, like C right here. This is a vacuole probably, or a lysosome. I can't tell which one is which. This one right here, I think, is a Golgi apparatus because it's going to start forming around things. 6, tell me some of the key differences between a plant and animal. That literally came right out of our notes on a slide. Or here's the one I really want to get to. Give me an everyday life analogy for a mitochondria, a vacuole, an endoplasmic reticulum, or a Golgi apparatus. Okay. So we've kind of already talked about some of those ones here. So, okay, that's all I've got. So, have a great long weekend.
You too.
What was that? I don't care. Your life's not dictated by a bell. Well, I mean, yeah, you guys can go if you want. I'm just saying, I'm done. So. I do, yeah. You want to look it over?
Yeah.
Give me a sec here. Let me stop recording though.